So chapter 6 was kind of complicated and we didn't really talk about it very much, so I wanted to go over it with you guys again. And it starts off with cells and the flow of energy. So it starts off by defining energy as the ability to do work or bring about a change. So energy is anything that gets the job done. And organic nutrients provide living things with this energy, and they capture this energy through the sunlight. So organic nutrients are things like glucose and ATP and everything like that that store energy captured from the sunlight to provide us with energy. So it comes from the sun, goes into the producer. We can't do this. We need plants to do it. And we get a carbohydrate which stores energy out of it. So there's a couple different forms of energy. Kinetic and potential are the two main ones. Kinetic energy is energy of motion. So anything moving has energy, and that's kinetic energy. And then when things are not moving, they can store energy as potential energy. So this is stored energy whose capacity to accomplish work is not being used at that moment. So there's energy to be used later, but it's not being used right then. Food has potential energy because we eat it, and then later there's energy stored in the chemical bonds, which later will be released to power things inside our bodies. Food is specifically called chemical energy because it's composed of organic molecules such as carbohydrates, proteins, and fats. And then the last kind of energy is mechanical energy, which is kinetic energy that comes from the potential energy stored in chemical energy. So basically, we have chemical energy stored as food, and then as we release that energy, we use it for mechanical energy. So here, for example, there's solar energy, and it gets stored into chemical energy through photosynthesis. So now we have chemical energy. This moose is going to eat a plant, it's going to get some chemical energy, and then it's going to break the bonds that are stored in the carbohydrates, and then it's going to use the chemical energy, release it, and use it as mechanical energy to move. Now there's two laws of thermodynamics which regulate the flow of energy through living things. Because energy is always moving through living things. So only a little bit is actually used. The rest is always lost as heat. And energy from the sun, mostly we lose it as heat, but we're still able to capture enough to power all living things. So the laws of thermodynamics explain how and why this energy flows. So the first law of thermodynamics is called the law of conservation of energy. So energy cannot be created or destroyed, but it can be changed from one form to another. So plants don't create energy, but they change it from solar energy to chemical energy. And that moose isn't creating any energy, but it's changing the chemical energy into kinetic energy as it walks. So that is the first law of thermodynamics. The second law of thermodynamics says that energy cannot be changed from one form to another without a loss of usable energy. So as we change from mechanical energy to from chemical energy to mechanical energy, or from solar energy to chemical energy, we're storing energy, but we are losing some of it as heat. And when we lose that as heat, it's no longer usable. And no process is actually 100% efficient. Cars that we use, the energy that they use is stored as gasoline, and they're about 20 to 30% efficient. And cells are even better than that at 40% efficient. So the rest of that 60% energy is lost as heat inside of our cells. So there's a different way to state the second law, which says that every reaction increases the disorder of the universe. So we measured this disorder in something called entropy, which is a relative amount of disorganization. So the entropy is how organized is the universe. So as glucose breaks into CO2 and H2O over time, so here we have glucose, and it breaks into carbon dioxide and water, it becomes more organized. Oh, that happens because glucose is more organized and less stable than carbon dioxide and water. So here we have glucose. It's more organized. The things are put together better. There's more potential energy, but it's less stable because it has less entropy. And then as it breaks apart into into water and carbon dioxide, it's less organized, has less potential energy, but it is more stable because it has more entropy. This also can explain why hydrogen ions move across a membrane. When they are distributed randomly, the entropy has increased. And everything in the universe is always working to increase entropy. So here we have all of the hydrogen ions on this side of the membrane, but you know that they'll go and they'll move through this channel protein and they'll spread across, and that's because over here they're much more organized, they have more potential energy, but they're less stable, and as they move across, they become less organized, less potential energy, and are now more stable because they have more entropy. Think of it this way. So a neat room is very organized, but it's much less stable than a messy room. So it's maintaining order requires an input of energy, which is why all life needs a constant input of energy from the sun. So maintaining order inside a room requires a constant input of energy. You need to constantly be putting things back where they belong, which is why it's less stable. It's much easier to get a messy room than to get a clean room. You need to put in energy to keep a room clean. You do not need energy to make a room messy. The next section is called metabolic reactions and energy transformations. So you guys know that metabolism is the sum total of all chemical reactions in a cell, and that all living things maintain structure and function through chemical reactions. So we know that reactants go in and products go out. After a chemical reaction has occurred, the amount of energy left to do work is called free energy. So after this reaction has happened, there's going to be some energy left hanging out, and that's called free energy. The letter we use to describe energy is G. So the change in free energy, delta G, delta means change, G means free energy, is measured by subtracting the free energy content of the reactants from the products. So negative delta G means products have less free energy than reactants, and reactions will occur spontaneously. So your metabolism includes both spontaneous and energy requiring reactions, and we have names for these. Exergonic reactions, X means out. Exergonic reactions are spontaneous and release energy. They release energy out, they are exergonic. Endergonic reactions require the input of energy to occur. Endergonic, they take in energy. And for endergonic reactions to occur, they must be coupled with exergonic reactions, such as the breakdown of ATP into ADP and an inorganic phosphate. So exergonic releases energy, endergonic takes in energy, and this energy we call free energy. So here we have ATP. So the more activity that your cells are doing, the more ATP is required because it provides energy for everything. So it is the common and universal energy currency, so everything uses ATP. And the reason for that is because it can be used in so many different reactions. So ATP itself is a nucleotide, so it's a nucleic acid. It's made of the base adenine, which is right here on the side, adenine, a 5-carbon sugar, which in this case is ribose, and 3 phosphates. So the 3 phosphates repel each other. They don't want to be near each other, so they create instability and potential energy. And it's high energy. This molecule is high energy because we can easily release this phosphate and release energy. So here we have ATP. It's unstable and has high potential energy. And then as we release this ATP, this extra phosphate, it is an endergonic reaction. As we release this phosphate, we are hydrolyzing it, and we are releasing previously stored energy. And we are allowing the change in free energy to do work and drive other processes. So this change in free energy is what is making or moving those hydrogen ions across the membrane or doing any work inside your body. The hydrolysis of ATP, we're releasing free energy. So going from ATP to ADP has negative delta G. And examples of this are protein synthesis, nerve conduction, muscle contraction, anything like that. So this is anything that's using ATP. Then, now we have adenosine diphosphate, which is the same thing, but now we have one less phosphate, and this is much more stable and has a lower potential energy than ATP. So we can't use ADP to do work. However, we can reattach this phosphate over here in an exergonic reaction. No, that is an endergonic reaction. Going this way is an endergonic reaction. So creating ATP up here from ADP and phosphate requires the input of energy from other sources. So this is where we need cellular respiration to make ATP, and we get that energy from glucose. This has a positive delta G because we are absorbing energy. And an example of this is cellular respiration. So, a coupled reaction is the use of an exergonic reaction to power an endergonic reaction. So exergonic reactions release energy and endergonic reactions absorb them. So if we couple an exergonic reaction with an endergonic reaction, we can use the exergonic one to power the endergonic one. So for example, we can use the exergonic reaction of ATP to ADP and phosphate to power anything else, anything that requires energy. So the energy released from ATP can power this reaction down here. So now let's talk about metabolic pathways and enzymes and how these work together. So metabolic reactions couldn't take place without enzymes, which is a protein molecule that functions to speed up a chemical reaction without itself being affected. So the enzyme doesn't change as it speeds up a reaction. These allow reactions to occur under mild conditions. They regulate metabolism, and they eliminate non-specific side reactions. So while most enzymes are proteins, not all of them are. Some are ribozymes, which are made up of RNA molecules that act as catalysts as well. And so chemical reactions occur not randomly, but are part of a pathway. So you guys know this, we've talked about different pathways, but they are part of intentional chemical pathways. And because of this, they have lots of many different steps, and each step is being catalyzed by a very specific enzyme. And so with intermediates, we can have different pathways interact with each other. So an example of this is glucose being broken down and forming ATP. So if it was one step, it would either be all or nothing. But we know that it's not all or nothing, because if there is oxygen, we will get all of our ATP, but if there's not oxygen, the pathway goes in a different direction and performs anaerobic respiration or fermentation, where we get less ATP. So different pathways can interact with intermediates. We can also release energy slowly. So if we are breaking down a starch molecule, we can, with all the intermediates, we can release energy slowly instead of getting it all in one big burst. And then we can also regulate each step, since each step requires a different enzyme. So we can regulate the pathway in many different locations. So these enzymes don't actually react unless they're activated in some way. And in a lab, we use heat to get molecules to react. And this heat, in this case, is what's called activation energy. So the energy that's required to activate the enzyme is called activation energy. And the reason that there is activation energy is because we don't want reactions to constantly be happening. We want them to happen when we want them to happen. And so this right here is the activation energy required for a reaction to occur. And enzymes are capable of lowering this activation energy by bringing reactants together. So heat speeds up molecules moving and brings them together quicker. That's how heat provides the activation energy. Enzymes actually take the two molecules, bring them physically together, and allow the reactions to happen. In this case, the reactants are called substrates, and the substrate fits perfectly into the active site of the enzyme. So we call this the lock and key molecule, because there's only one substrate for every enzyme. And as the substrate binds to the active site, the enzyme will change shape just a little bit to accommodate the substrate perfectly. So we call this the induced fit model. As it binds, it changes. As the substrate binds, the active site changes just a little bit to perfectly fit around the substrate. Enzymes are named after their substrate. So for example, lactase breaks down lactose. So that's how you can tell. And here we have two different ways enzymes work. They can either degrade molecules, meaning here's the substrate, fits into the active site of the enzyme, then the enzyme breaks it into two different pieces and the enzyme leaves unchanged, or we can have a synthesis enzyme. So we have two substrates, binds to the active site of the enzyme, binds them together, so now we have one product, and the enzyme again leaves unchanged.